Hello, everyone, and welcome to our last Retina web seminar of the year. 2020 has been quite difficult for us all, but somehow in these strange times, we all together managed to tame a new media, and I have to say that I am quite impressed by its increasing popularity. So first, I'd like to thank all participants who appeared on this platform since last April, and I'm really looking forward to receive new talkers for the upcoming years. Once again, we would like to thank Tim Pogels and Panos Vozelos for setting up the worldwide Neuro platform. I know I repeat myself a lot, but please do take the time to check the website. I'm sure you will find topics that will be of interest for you. Also, for the 2021 series, if yourself you would like to share some of your recent works or participate in a discussion, please get in contact with us. That being said, today I'm very happy to receive Wei Li from NIH, based in Bethesda. Dr. Li received his medical degree in 1997 from, sorry for my pronunciation, Zhejiang University School of Medicine in China, and his PhD in neuroscience in 2003 from the University of Texas at Houston. There, he studied the organization of reciprocal feedback synapse at the axon terminal of the retinal vacuolar cell in Stephen Massey's lab. He then worked with Stephen Iveres at Northwestern University in Chicago, where he investigated synaptic connection between photoreceptors and vacuolar cells in mammalian retina. Dr. Li is now a principal investigator of the unit on retinal neurophysiology. His unit uses a variety of physiological and anatomical techniques to explore retinal synapses and circuits and their function in vision. So good afternoon to our European audience, and good morning, Wei.
Well, thank you, Maxime, for the nice introduction. Can you hear me okay?
Yep.
Okay, I will start first sharing my screen. Sharing.
Perfect.
Is it good?
Looks pretty.
Okay, thanks. Well, thanks, Maxime and sorry, Tom, for the invitation. And it's great to see colleagues, friends online. And I think I still remember the first talk of this series is at Jeff Diamond, I believe. That's the first kind of online gathering for our community. And I was so excited to see each familiar names that popping up in the chat box and greeting from all over the world. So it was truly an enlightening moment in this dark period of pandemic. So thank you, Tom, and your teams for organizing this, and I'm honored to present here today. As many of you know, my lab study Retina as a model system to understand different aspects of the central nervous system, including development, physiology, metabolism, and disease, etc. So today I would like to share with you some unpublished work on what I think is a prime example of how cellular structures in sensory neurons that could function to maximize sensory input. And the work is done pretty much single-handedly by a very talented staff scientist, John Ball, in my lab. And I really look forward to feedbacks and comments from all of you. And for those of you who happened to attend the Neuro Zoom talk a couple of weeks ago, organized by Alan Gittler and Zilong Xu, you may take a coffee break for 20 minutes since I present the same first half of that talk. So I guess for this audience, I don't need to, but since the YouTube gonna live online for a while, just for the non-retina aficionados, a very brief introduction of the anatomy. So our vision emerges from the collaboration between the eye and the brain, and the retina, which is a thin layer of neural tissue in the back of the eye, really is the place where the brain first sees lights. And for us human, we have this unique structure in the retina called fovea region, which is, as you know, is very critical for the daylight vision, color vision, high resolution visual processing. If we take a cross section of the retina and look at the structure as drawn beautifully by Ramani Kahal here, you see photoreceptors, including rods for the night vision and cones for daylight vision, and also how they relay the information through interneurons, bipolar cell in this case, to output neuron, the ganglion cell. But really a cardinal or essential feature of the retina structure is that it is inverted, and such that light has to pass through all the structures of the retina to reach the outer segment of photoreceptor where light sensing molecules are housed. So this particular inverted feature of the retina structure, along with, of course, the overall sophisticated eye structure, has been really at the center of debate about evolution for many years, dating back to Charles Darwin, who wrote it in on the Origin of Species, that he said the idea of something like the eye, I quote, could have been formed by natural selection seems, I freely confess, absurd in the highest degree. And of course, he made this comment in awe of the power of natural selection. However, it's often being misquoted as evidence for the divine. Such, complex but precise structure can only be created by a wise or benevolent creator. So the evolution biologists, on the other hand, argued later that, in fact, the structure of the retina of the eye is not perfect, and like many parts of the body. And those imperfection really reflects the stepwise process of evolution rather than a one-time designing process. And as example, in this book, Darwin's Gifted to Science and Religion by Francis Ayala, a famous evolution biologist, argued that, again, the retina, the inverted structure of retina, as example of this imperfection, and he even called it dysfunctional. Now, this time, in fact, it was the creationists argued that with a lot of actually scientific sound evidence that some of those imperfections indeed are not, but by design to help the visual processing. I'm pretty happy now to see such debate these days, because at least it's based on the same set of facts rather than having one side arguing with alternative facts. But that is not to say that I'm here to support creationist and this evolution. But at least for the retina structure, I would humbly argue that some of those seemingly flawed structure may be beneficial and reflect evolutionary adaptations. That reminds me a bit of what's recently have a very lively discussion on Twitter. I'm imagining Tom smiling about whether the retina structure is optimal, near optimal, or just good enough. So with that introduction, what the question that we are looking at in this project really is illustrated here. So again, I show you a kind of cross-section of part of retina, focusing on the really the photoreceptor. What we, or people known for quite some time, is that the mitochondria, here stained with antibody against COX-1, I believe, the mitochondria in the cone photoreceptor, and this is ground square retina, so mostly cones, are largely concentrated in the upper portion of inner segment, the so-called ellipsoid region, and right before the outer segment. And that means, again, the light has to go through all the structure, and particularly at the last sort of huddle to reach the outer segment for reception. And the question, of course, is why this is happening. And one could argue that outer segment needs a lot of energy, and certainly a valid point. But the particular organization of those mitochondria is still an interesting phenomena. And this is very relevant to human vision because, as we all know, and I mentioned earlier, the fovea is the most critical part of our anatomy structure in the retina for our vision. And it's formed such that most of the retina structures got pushed away to give way to the light to pass through to be received. However, this inner segment, ellipsoid region where those mitochondria resides, remain at even the very center of the fovea. So again, it's a intriguing structure arrangement. Now, this is relevant because we know that mitochondria has slightly higher reflection index. And although if they compared to the cytosol by a small amount, but we know that the small difference in reflection index could make a huge difference. As example, optic fiber are designed using even smaller difference between the center core and the surrounding clay. So potentially, and they could affect the optics. Now, this is also not a novel or outlandish idea. The cellular or intracellular structure play optical roles in the retina has been previously reported. There's a lot of work done on Mueller cell, potentially acting like optical fiber. Also beautiful work looking at nuclear chromatin structure of raw photoreceptors, and by modeling and comparative anatomy of different species, really showing a role of raw nuclear in light passage. So in light of the terms gene sharing in developmental biology, I would argue it's sort of the organelle sharing, if you will, to use the organelle for multiple functions. So here we're really looking at a couple of possibilities. One, that maybe mitochondria are there for the purpose of energy. However, it's not good for light passage. It could scattering the light and reduce the number of photons delivered to the outer segment. Or, as some people has speculated, maybe they're good there. They could potentially say acting like a fiber optics and concentrating the light delivered to the outer segment. So ideally, you wanted to visualize that to see what is light path through the structure look like. And that's pretty much our plan. So we wanted to look as the light go through and use a microscope objective in lieu of the outer segment and see what the outer segment sees, essentially. So as you can see, if we do that, and there's a lot of structure in the retina, so we really cannot attribute this optical effect to the mitochondrial bundle in the inner segment. So a near optimal condition will be if we could have a single layer of cone photoreceptor and shine the light through and see what come out of the other side. An optimal condition will be that we could even get rid of the nuclei, so only look at the inner segment or mitochondrial bundle. And of course, that's kind of an ideal dream condition that we couldn't dare to dream about when we started. But we went ahead anyway, and this is the animal model we choose. As many of you know, we work on the 13-line ground squirrel, and it's very unique as this cone-dominated species, and therefore they have big, large cones to image from. And also, a second feature is that they hibernate in the winter time, and so the retina is particularly hardy to experimental manipulations. So that's good for a long period of imaging experiment. And also it comes with some additional advantageous features, which will become clear in the later part of the talk. So here's what we're doing, and we take a piece of ground squirrel retina and embed it to agarose, and we cut horizontal slice through to with the hope to get a single layer of cone photoreceptors. Again, just to remind you the structure here, the inner segment where the mitochondria is and the nuclei, and this is synaptic terminal. But what we found, interestingly, intriguingly, and perhaps with some luck, that we found a lot of our preparations, we couldn't even see the nuclei, that we wound up having just a layer of the inner segment. So I think what happened is that the section process really break the cell in some cases at the narrowest joint between the inner segment and the nuclei, and the membrane reseal. So of course, this is a pretty ideal preparation that one could imagine. But you would argue, you would worry whether the cell or mitochondria is still in good shape without intact cell structure. So we use a TMRE, which is live mitochondria labeling. It's essentially a positively charged dye, so only when mitochondria maintain its very negative membrane potential, you will see the fluorescence. So it gives us the position of the mitochondria, but also tell us the healthy status of mitochondria. So this is a real vibrotome section, and you can see the staining of patches of cones with live mitochondria. And then we put it on in a microscope. We modify it such that we can do transmitted light and fluorescence light confocal imaging simultaneously. So when you do that, and you can do a kind of standard image with a stack of layers, this is the fluorescence image, as you can see, labeled with TMRE. You can take a thin slice and look in the vertical projection, and you see very similar structure as you will see in the retina section, mitochondria bundle, that is. Now, if we image the transmitted light simultaneously, then you can see once the light passed through this mitochondrial structure, it becomes quite concentrated. And if there's no mitochondrial structure, then you don't have that. So this is quite striking. And I wanted to emphasize this is a real image. This is not a diagram showing the light passage. So this is real light distribution after the pass through the structure. Now, if we look at it, the structure in more detail compared to the anatomy of the cone, inner segment, outer segment, it becomes quite obvious that the part that we're like concentrated is where the presumptive outer segment will be. I want to stress that in most of our preparations, the outer segment is gone as we strip away the RPE. So we're really looking at what's like it looked like as it passed through the outer segment, inner segment junction here. And the diameter of this concentrated light beam is about 1.5 microns, close to the diameter of the outer segment. So we quantify that, John quantified that by measuring at individual layers the intensity of the both mitochondria and the light signal, and plotted against the distance. We kind of define the concentration of light or focusing as the transmitted light gain in this presumptive 1.5 micron diameter outer segment, and compared to the background. So give you this distribution curve. And if the curve move up, right, or leftwards, that means more light potentially in the outer segment. And if it move downward, that means perhaps less light in the outer segment. So, so this is all good. At least we see that this is not bad. We didn't see a reduce of the light or scattering due to scattering, but rather we see a concentration of light in the position where the outer segment will be. We're not sure whether this is fiber optics or not, but the light concentration seems to be quite obvious. Now, of course, the question is that because of a mitochondria? Because people have been looking at that for quite some time, and particularly the shape of cones, the membrane of structure of cones could potentially play a role in this light guiding or concentration behavior. So ideally, you wanted to change the structure of mitochondria, and then you observe whether that changed focusing. That's why the hibernation become handy. So it turns out this mitochondrial bundle structure changes when they hibernate. As you can see by eye, you can see how this awake condition, these mitochondria is organized in like a spaghetti bundle, whereas in a hibernating condition, they lost this very organized parallel structure. And this is achieved by zero Brock phase reconstruction. And I wanted to mention and stress that Ben Sedgwick in Joe Carroll's lab did a very similar work and beautifully together with adaptive optics and published last year. This is a very good because for those small amount sample reconstruction, you always wonder this is just a fluke or it's a real. So we're happy to see the convergence of the result. So we quantified the structural change by looking at individual mitochondria, their angle or their axis difference to the main axis of the photoreceptor. And that difference distribution, as you can see for the awake, is very narrow and close to zero, indicating they're almost parallel, aligned along the axis of cone photoreceptor. Whereas the hibernating one, the distribution become wider, indicating that they lost this kind of spatial arrangement. Now the question, of course, is with that different structure, although subtle, will that lead to a change in the focusing phenomena we observed? So John did experiment in the awake versus hibernating condition. As you can see, there's quite a bit of variation between both groups, but by and large, the active one, the awake one, compared to the hibernating one, they have better focusing or concentration of light. And he did bootstrap statistics to make sure that they're statistically different. We'll talk a little bit about these variation. In general, we observed that what tissue has more intact structure, like in a more complete patch, has better focusing than the one with individual cells around. But as you can see, this change, although significant, is still relatively subtle. So what we really wanted to know is, without those mitochondria there, how does that impact focusing or light concentration? So to that, we had to turn to modeling of this mitochondrial bundle, its impact to light passage. And cone optic modeling has had a long history. And back in 50s, people will stick a large styrofoam through the window and have microwave pass through to model the cone structure impact on light passage. And this plan didn't go well with the building folks, so we have to restrict ourselves to the computational model. And so that's what John chose to do. He used this finite difference time domain electromagnetic simulation. Basically, treat light as electromagnetic wave and calculate in a grid, 3D grid of structure with 10 nanometer resolution, calculate for each individual one of those voxel, the energy as this monochromatic wave pass through the structure, and look at the energy distribution at the other side. So what we observe, interestingly, is that once the wave goes through the structure, it also concentrates in a structure that very similar to what we observed experimentally. So with the modeling, now what you can do is basically separate the role or contribution of mitochondrial bundle and other structure like cell membrane, for instance. So here's what he did, John did, and showing that in the intact structure with the cell membrane and mitochondrial bundle, cell membrane alone, cone inner segment cell membrane alone, and mitochondria alone. And even by eye, you can see that the mitochondria alone did quite well in terms of concentrating light to the presumptive outer segment region. Whereas in the without mitochondria, you still have light focusing or concentrating, but it will largely miss the outer segment. And this is quantified here. Again, the mitochondria alone did quite a good job concentrating the light, whereas the membrane alone is not good enough. Now, if we look at the hibernating condition, interestingly, that the mitochondria alone also not as good as the control condition in terms of the light concentration, indicating really that the parallel structure of those mitochondria is important for this function. So this, I think, really is the one of the essential message I wanted to deliver, that mitochondria play a quite important role in the way that they're concentrating the light. And with modeling, John was able to play with different structures and options and shape and forms and largely maintain the volume, but change those parameters and see what's the best. And two obvious winner is actually one of those spaghetti bundles, as we see in ground squirrel cones, as well as in the human cones. And interestingly, another winner is just big, giant single mitochondria. And this is also intriguingly observed in treeshore retina, that you have this just, some of their cones has this giant mitochondria sister, and it's actually termed lens mitochondria. And people been speculating and play a bit of that, you know, what potential optical role they might be in, and it's called a lens mitochondria. So if it's, that really got us thinking that maybe this bundle of mitochondria perhaps behave like a micro lens sitting there. And if it is a lens, of course, you can form an image. And that's what John set out to test. So what he does is put a slit along the light path and image what on the other side of the photoreceptor. And then he rotated the slit 90 degrees and took another image and overlaid those. And interestingly, indeed, where you have an intact bundle of mitochondria, you can see the images. I can zoom in one for you, and the two image overlay, you can see the cross, and the orientation of the cross is in fact flipped, and that's reflecting the feature of lens. So what I show you is so far mitochondria seems to be quite good in concentrating light, and perhaps they're acting more like a micro lens than optical fiber. And last couple weeks ago, when I gave this online talk, I had a very helpful discussion with Daniel Planker. And he pointed out the issue that why the concentration of light is so limited and it's not spreading out. And in fact, when I posed a question to John, and both John and Daniel gave me the exactly same diagram showing that what we, at least what John considered a scenario of a ball lens. And I think in this case, we're perhaps looking at more of a elliptic ball lens than the spherical one, of course. So you have kind of a spread focal point and narrow concentration of light. So just to summarize so far, what I show you, I hope you'll be convinced that this bundle of mitochondria in cones may act as microlens to concentrate light and deliver to the outer segment. And this elongated parallel structure seems to matter. Now, if they're lens, then there's some of the feature that we should consider, in particular related to the direction of the light passage. So if we consider the light incidence of light angle that is not in parallel or aligned with the axis of the cone, but at a slight angle, and that will it means that at the presumptive outer segment, some of the light will miss the outer segment. And therefore, there will be a kind of angle dependent of light delivery. And that is very much related to a famous Stiles-Crawford effect that discovered back in 30s by Stiles and Crawford, who discovered that the same light get into your eye at a different angle are perceived differently, and with a very sharp decline of sensitivity in relationship to the angle of incidence of light. And traditionally, this phenomena has been largely explained by the light guiding feature of the photoreceptor cone structure, shape particularly, and we'll talk a little bit about that. Anyhow, as summarized by Jared Westheimer in his beautiful review in 2008, that this Stiles-Crawford effect really is one of the a great discovery in visual science, and its explanation remain challenged or not fully explained. One of the anatomical feature I think is relevant here that we see, and also previous been reported and stressed, that is the outer segment seems to be always in line with the inner segment in a normal condition. This is what you will see for the ground scroll retina when we keep the RPE, so to keep the whole structure intact. You also will see that in the monkey retina as well. I will mention that there's some beautiful work coming out. In fact, this alignment is regulated, but I don't want to steal the thunder. There's interesting work coming out very soon. But this fact, the inner segment, outer segment aligned, indicating that if your light's coming in at a slightly different angle and it behave, the structure behave like a micro lens, the outcome might be that your outer segment will miss a lot of the photons or light. So John modeled that by changing the angle of the incidence of light and looking at the amount of light in the presumptive outer segment. And he calculated and plotted the same way that people will measure Stiles-Crawford effect. And what he sees is that indeed you have very sharp decline of the amount of light with a rho, a parameter. You can think about it as almost like a direction index for the light reception. And this is actually very close to what initially measured. I believe this is one of the actually Stiles' own eye measurement. The normal people, you will have a rule about 0.05 to 0.06 and quite similar. But if we look at the condition without the bundle of mitochondria, you will have a much kind of a impaired or reduced rule. So this is all interesting. So indicating that this micro lens feature could potentially explain, at least in part, the Stiles-Crawford effect, the kind of the angular dependence of this light reception. Then, of course, this is modeling, and we realized that in our experimental data, in fact, because of the experimental manipulation, we have a scenario that not all the mitochondria are aligned well as in vivo. So some of them, they tilted certain angles. So in fact, we could take advantage of that, measuring the angle of mitochondria and by that way, the axis of the photoreceptor and outer segment. and then look at the relationship of this angle to the amount of light that you will receive in the presumptive outer segment region. And when we do that, interestingly, we can see that indeed there just like the modeling predicted, there is a very sharp decline. And John separated those preparations into two categories, one with quite good intact patches, one with more sparse conditions. And clearly the intact condition, perhaps more reflecting the in vivo scenario, you have a better focusing and you have a better angular dependence, a rule number that's close to a fairly normal condition. And in fact, if we look at the relationship between this rule, the angular dependence, and the focusing power, there's a very good linear relationship indicating that that once you have lost the focusing, then you will decrease or damage this angular dependence. So just to quickly summarize, I hope I can convince you that this bundles of mitochondria encode photoreceptor right in front of the outer segment, could acting like a micro lens, and to that facilitate the delivery of light to the outer segment. In a way, you can think about it as enhanced coupling between inner segment and outer segment. And this mitochondrial bundle arrangement is important. And this phenomena could potentially contribute quite significantly to the Crawford effect. So I wanted to go back to the fovea scenario a little bit and talk about potential implications. The question is, of course, what we observed in ground squirrel, how it is relevant to our human vision. And I took this beautiful histology images from the book chapter by the late Anita Hendrickson in this book of macular degeneration. And a lot of other people, Kristen Kircher, Rachel Wan, and Steve Massey et al., they all have similar data. But this, in this book, Anita really demonstrates beautifully the structure of fovea region. And I just denote the areas as she did in the book. So as you can see, so this is two subjects actually, one younger one, older one. As you can see, apart from the very center, what she called, or people call it foveola, where you have very slender, skinny inner segment and outer segment. And once you hit the fovea slope, and the inner segment structure, and certainly in the parafovea region, the inner segment structure become much thicker and very similar to what we see in the ground squirrel retina, in fact. So here, as you can see, the fovea region. So I think in the outer segment of foveolar region, where you have very thin inner segment, interestingly, also the mitochondrial density is not as high. Perhaps the structure or the shape of the cone play a more important role in this Crawford effect, as very nicely modeled by Midway and Sinchich recently, and a lot of work in previously. But interestingly, Jared Westheimer pointed out in his 1967 paper, where he looked at the Crawford effect at different regions and pointed out, in fact, the parafovea region, you had the most highest Crawford effect. And the foveola, the center, really very center, is not as good. So what we think it is, what we describe here is have more relevance to the parafovea of human than perhaps in the central, very center part of fovea. So another thing I wanted to point to is there's other stuff in front of the outer segment in other retina structures. It is, of course, well documented, such as this nice review by Jacobo et al. For the turtle retina, you have obviously the oil droplet, and this recent work in a bird, the flycatcher, and they have, in addition to oil droplets, some very peculiar structure. And later through EM, they found, in fact, a giant mitochondria and was oil droplet around. And more recent work by from Tom Baden's lab, Takeshi Yoshimoto beautifully pointed out that the kind of a prefoveal-like strike zone of the zebrafish larva photoreceptor, where they have the specialized structure, including enhanced mitochondria right in front of the outer segment for potentially enhancing the ability to see prey. So at this point, I can't help to draw your attention to the compound eyes, which we traditionally think is very different from vertebrate eyes, of course. But in compound eyes, for each photoreceptor, you have their own little lens to facilitate the delivery of photon. And if you compare to the vertebrate eyes, and it's not too surprising that for each photoreceptor, there might be optimized the optical structure to facilitate the delivery of photons. So perhaps reflecting conversion evolution. So at the very end, I would just mention a bit on potential applications. Because we know that many of the retina degenerations involve photoreceptor mitochondria change. And if we link this Crawford effect to the structure change of mitochondria, and this, which is a psychophysical measurement, perhaps can be used for diagnose tools to identify early mitochondrial structural change. And in fact, looking at the literature, people did find by Elliot Burson's group that Crawford effect changed quite significantly in early RP patients. And this, I think, is very much in line with what we observed, that the decline of this rule is closely linearly associated with the decline of the focusing power by the cone structure. So with that, I will stop and again acknowledge John really did a bulk of the work with help from Shan Chen, who recently retired. Shan did horizontal retina section, which is quite critical for the work. And we hope the reviewer won't ask for more experiment because Shan retired recently. And current lab, they all contributed a lot of discussions and former members and the founding source. With that, I'll stop and thank you for your attention, and I'll be happy to take questions.
Thank you, Wei. That was very interesting. I encourage people on YouTube to join us on a Zoom room. You will find the link on the description if you want to ask your question yourself. I don't see lots of questions, but I guess I have one from Tom. I think he's referring to the first part of your talk when you were describing the focusing effect of mitochondria on Optical. Is it wavelength selective?
It's a very interesting question. So let me go to, I might have a slide. So we haven't done extensively, but we did, John did a couple of experiments with different wavelengths of light. And I have to check on the modeling part, but with a limited amount of experiment, he does see a slight favor of green wavelengths, perhaps, over the short wavelengths and longer. So this is one set of experiment that taking multiple examples with different wavelengths of light, either in the around 450 or around 520-ish compared to the red. So there is a slight chromatic selection, if you will, and that's also observed, I think, reported for the Crawford effect.
Thank you. I have a question from Brian Clark. So really, really cool stuff. Does the degree of mitochondrial packing or organization affect cone outer segment length across various species?
The relationship between the mitochondrial bundle and the cone outer segment, we have not looked systematically or in a comparative anatomy fashion, but as we actually discussed with Maxim and Tom earlier, that there's appear to be a huge variation across species. Some with much longer outer segment and even the inner segment bundle structure differ, but systematically, if there's a correlation, we haven't looked very carefully yet.
We have Karola Jovanovic with us today. So nice talk. How will transparent all droplet, so in birds, for example, incorporate into these pictures? They also apparently act concentrating light, so will this render them redundant?
Well, as many of us know that our droplet, the main function is chromatic filtering, and I think there's some nice modeling work done by Joe Cobb and Nicholas Robert, and looking at this issue of chromatic filtering by oil droplets. So I think they might play a role, a similar role in focusing, but perhaps more in the as a chromatic filter.
So you cannot see the chat, so I'm just going to tell you that your former PI is congratulating you. We have a question from, from Miruna Binitegla, who says amazing work. Is there a similar function of the mitochondria in rods?
Good question. So as I forgot to mention, so just like the I think the foveolar cones that has maybe less dense of a mitochondrial bundle. Rods has less dense of mitochondrial bundle. And correlated to that, rods has been well documented, has less Crawford effect. So this at least is in line with that observation. So they might have a similar impact to the light focusing or concentrating, but maybe not as significant as the dense bundle that you would see in cones. We would be happy to model there's some very nice rod EM data out there from mouse. We'll be happy to look at it in model.
Sorry, other questions. From Nogavardi. So nice presentation.
Thanks.
Do you have an image of the light as it passes through the whole retina? How much more diffuse it is, if at all?
Well, yeah, that's also a very interesting question. So I believe John did some early experiment before we settled on the horizontal section, some light passage from the whole retina. I think you can see similar focusing qualitatively, but perhaps not as clear as what we show here. On the other hand, if you have more intact preparation with photoreceptor, including nuclear and intact structure, then the focusing is better. So possibly there's some degradation of light through the restive layer of the retina, but this part is critical as, on the flip side, also there could be other mechanism to facilitate to deliver light to that point, because even Mueller cell or nuclei stops before the inner segment. So either way, that the last part of this structure is the last hurdle that you have to go through to reach the outer segment. So the function of those is sort of a last relay for the whole process.
Thank you for that. I have one last question from Ulyss. Very nice work. Do you expect a similar effect in roads? Will this happen also in the transition between light and dark?
So I think I talked about the rods earlier, but the transition between that we have not looked, at least not through detailed EM structure analysis by light microscopy. Perhaps we don't see a significant change of arrangement of mitochondria, transition between light and dark and even night and day, but that hasn't been looked carefully at the EM reconstruction level.
Well, thank you for all that. Thanks for your questions. If you want to join us for an after talk room, I suggest you join us now. I will soon close the YouTube video, so please join us on Zoom now. Thanks, Wei, that was very interesting. Thanks for the audience to join us again. I hope we will see you again in January. We should restart those talk series in, let's say, mid-January. I hope so, and also on a bi-weekly basis. So I hope to see many of you there. I'm going to close the YouTube channel now, so please join us.
Thank you.
Thank you. While we're waiting, I have a stupid question to ask.
Yeah, oh, no stupid question. Go ahead.
What would be the point of for